Today I will be talking to you about serendipity in drug discovery, and specifically penicillin. But to do this first, you have to ask, what is serendipity? Serendipity is simply the accidental discovery of something fortuitous. Serendipity has played a very important role in the discovery of many drugs used today. There are so many drugs that were discovered by accident, and a few examples are penicillin, which acts as an antibiotic, warfarin, which acts as an anticoagulant, and chlorpromazine, which is an antipsychotic drug. As I said, penicillin is an antibiotic. An antibiotic is defined as substances which are derived from bacterial sources that are microorganisms that control the growth of or kill other bacteria. Penicillin was serendipitously discovered in 1928 by Alexander Fleming, who was a Scottish scientist. His discovery led to major changes in the pharmaceutical industry. Fleming was undergoing research on influenza when he noticed that one of his Staphylococcus culture plates had become contaminated. This plate was dotted with colonies which had developed a blob of mold that created a bacteria-free circle. Fleming then decided to isolate that mold in the culture, and he realized that it produced a substance which was capable of destroying a wide range of harmful bacteria such as meningococcus and streptococcus. Fleming named the antibacterial substance penicillin after Penicillin notaeum. This is the chemical structure of penicillin. There are two amide groups, a carboxylic ion, and a thioether. Penicillin also has three chiral centers. Next we move on to how do antibiotics work. Antibiotics work by affecting the things that bacterial cells have, but human cells don't. This is generally the cell wall. Human cells do not have cell walls, while many types of bacteria do. A lot of antibiotics work by attacking the cell wall like penicillin. These are drugs that specialize in preventing the bacteria from synthesizing a molecule in the cell wall called peptidoglycan, which provides the wall with the strength it needs to survive in the human body. Antibiotics also work by affecting the ribosome, which is the cell's protein building site. Ribosomes are known to build proteins on both bacteria and human cells. However, there are obviously differences between bacterial and human ribosomes. Due to the fact that proteins do all the cell's work, a bacterium that cannot build proteins cannot survive. An example is erythromycin, which is commonly used to treat respiratory and skin infections. Other antibiotics prevent successful DNA replication of bacteria by causing the strands of their DNA to break and preventing them from being repaired. An example is ciprofloxacin and levofloxacin, which are both used to treat infections like bronchitis and pneumonia. Finally, we look at the impact of penicillin on the world. Due to the introduction of penicillin, other industries began to develop other antibiotics. The development of meprobamate, which is the first anxiolytic drug introduced into clinical practice, was also the result of a serendipitous observation in the course of this research. In 1942, the first patient was successfully treated for streptococcal septicemia in the United States. However, due to the high demand of penicillin, the supply was limited. Penicillin also helped reduce the number of deaths and amputations of troops during World War II. Penicillin has made such a huge impact on the world today by changing the world of medicine enormously. Infections that were previously severe, like bacterial endocarditis, bacterial meningitis, and pneumococcal pneumonia, can easily be treated today with penicillin.